All right, so the first phyla of animals that we're going to cover are Porifera, and that's fancy sciencey talk for sponges. There are certain characteristics that all sponges have in common. For instance, all sponges are aquatic. You're going to find them living in water, typically salt water. All sponges have pores. These are openings that allow water to come in to the sponge, and that's also going to bring with it food for the sponge to eat. Sponges have two germ layers, and they don't have mesoderm, so they only have the endoderm and ectoderm. They don't have very organized tissues at all. They don't have organs, but they do have specialized cells, and those specialized cells are going to work together to accomplish goals of keeping the sponge alive. But even though these specialized cells are working together, they do not form tissues. All sponges are asymmetrical. They have no specific symmetry. The adult sponges are sessile, and that means they're attached and are non-moving. In between the endoderm and the ectoderm is a mesenchyme. It's a non-living layer, so that's why it's only two layers. The mesoderm isn't there. The mesenchyme is non-living. And finally, all sponges have spicules, and these provide a support like a skeletal system, but they're not made of bone. And we'll talk more about what they are made of coming up, but it's non-living. All right, so let's take a look at these two pictures. First, this picture in the upper left-hand corner is a picture of a sponge, and it's zoomed in so you can see, like, here's a pore, and here's a pore, and here's another. These are in-current pores that allows water and other nutrients to flow into the sponge. On the bottom right here, we have a picture of an electron microscope image of a sponge, and what I want to point out to you are some spicules. You can see this here is a spicule. Notice the shape of it is that it's kind of like that peace sign without the circle, but when I make a circle around it, it kind of looks like a peace sign. You can see another one here and another one here, and you can see another one going here, and you can see a broken one, or maybe even the piece going down into there. So here's part of one. And remember, the spicule, its job is to act like a skeletal support system. So you're going to see them all over the place on a microscopic level inside the sponge. Sponges don't have organs and they don't have tissues, but they do have cells. And there's three main types of cells that exist in sponges that allow for them to live and survive. One type of cell, depending on the source that you see, we'll either call it a penocyte or a porocyte or a pore cell. These three names can be used interchangeably. I typically refer to them as pore cells. These are specialized cells that line the outer layer of the sponge and that regulate the passage of water and food into the sponge. These are the cells that make those pores we just saw in the last slide. A second type of cell are the mesenchyme cells, or sometimes you'll see them referred to as amoebocytes. Their job is to digest the food that comes in and distribute the nutrients to all the other cells. They also are the ones responsible for making the spicules, and remember, those spicules are what create the skeletal system for the sponge. And the last one are choanocytes, also called collar cells. And these collar cells are lining the inside of the sponge, and they create a current of water. And that current of water draws the food particles in. They have flagella that they use, and they like flip their tails. The flagella, remember, is shaped like a tail, just like on sperm. And the movement that the flagella makes allows for the water to circulate and bring the food to the choanocytes or the collar cells so that they can pass the food to the amoebocytes, and then the amoebocytes can pass the nutrients on to the porocytes and back to the collar cells. Here's a diagram of a sponge. On this side over here, we're looking at a sponge's exterior, and you can see all these black circles here and here. Those are the pores that are created by the pore cells. Now here over here, we're taking and cutting the sponge open so that we can see what it looks like inside. Now this is just a diagram of what the cells would look like, but keep in mind all this is cellular and so microscopic. You can see right here there's a purple circle, and that's meant to indicate a nucleus. So that means that this cell here on this side, and there's another cell on the other side, these are pore cells, and they can swell up and get smaller to allow more or less water to come in through the opening, that pore. We also see one here, and you can see a pore here, and as you can see, they go all around. Also, you can see lining the inside, we've got these collar cells. We'll come back to that because they blow them up on the other side. And then in between the two layers, you can see right here that bluish purple, that's another cell. That particular cell is an amoebocyte, and those are the ones that do the digesting. This internal cavity here is also known as the spongiceal, and we'll see that on the diagram in a minute. And then the water flows out the top here, and that's the osculum. Osculum is the singular version. Oscula is the plural. So most sponges will have one osculum. Some will have several oscula, depending on the type of sponge. Now let's come back to the collar cells. So here we have a blown up version of a collar cell. Again, we've got this purple circle here trying to tell you that it's a nucleus, so it is a cell. And here you have the flagellum. Remember, flagellum is singular, flagella is plural. That flagella will beat like a dog's tail back and forth, and by doing so, it creates a current in the water. And that current will find these little food particles like bacteria and draw the bacteria in towards the body of the collar cell. Then that collar cell can pass it along. You can see here this bluish purplish area. That is one of the amoebocytes, and that's the cell that's going to take the food and digest it. And then you can also see right here, that's one of those pore cells. The reason the collar cells here are called a collar is because they have that flagellum here, but they also have this like collar wrapped around here, and that's used to help catch the food so that it can get as much food out of the water as possible. And by doing that, it prevents the sponge from starving, which is going to be ideal for survival. This is another diagram showing basically the same thing. There's a couple additions that I do want to point out. You'll see that here they talk about the spongiceal, number 4, the spongiceal, and basically that's just this whole cavity in the middle. Here you also see the label, again, another labeled osculum. And the other thing that I want to point out to you is in yellow here, they've got, and all around, they're showing you spicules. And remember, they typically have that shape of like a peace sign, and my drawing skills are not great, but they will typically have this shape. And we'll look at those under a microscope soon. So remember taxonomy, we said kingdom, phylum, class, order, family, genus, species. So now we're going to talk about some classes of Porifera. So the kingdom's animal, the phylum is Porifera, and now we're going to talk about 3 classes. We're going to get more specific. The first one is Calcara. These are small sponges that typically hang on the ropes at docks, and they tend to live in more shallow water. They have spicules that are made of calcium carbonate, and that's one of the ways that we classify sponges is based on what their spicules are made of. Remember, the spicules are part of that skeletal system. Calcium carbonate is what we use to make chalk and Tums. So it's got a rigid structure, but it can be broken fairly easily. So these are delicate sponges. The next class is Hexactinella. These are commonly known as glass sponges. You find them in deep, deep water, and their spicules are made of silicon. Again, we classify sponges based on what their spicules are made of. And the last class of sponges that we're going to talk about are demospongae. And these are typically what you think of when you think of sponges. 95% of the diversity of sponges exist in this class. It is the most diverse class. And the spicules of these sponges are made of spongin fibers, which is a protein, very rigid protein, and silicon. We oftentimes don't think about sponges being living, so it's difficult to sometimes wrap our head around the fact that sponges are actually animals, and all animals have to eat. It's part of the characteristics of life to be able to obtain and use nutrients and energy, and part of that is metabolism, so they have to eat. What they eat, though, has to be smaller than they are, so typically they eat microscopic organisms such as bacteria, algae, and protists. These are all single-celled organisms that are constantly floating around in the water, and when you get out of the water at the ocean, at the beach, you're covered in them. You just don't even know it because you can't see them with the naked eye. Sponges eat by filter feeding. They use those collar cells with the flagella to create a current in the water that's inside the spongiocele, and that draws the food to the side of the sponge where the amoebocytes can capture the food, digest it, and then distribute the nutrients from the food to all the other cells. The water then exits through the oscula. Sponges being an animal means that they also need to be capable of reproduction. They do asexual reproduction, and they do that a couple different ways. One is through budding. This is where an external group of cells break off from an old sponge and go form a new sponge. They also have gemmules. These are an internal growth of cells. So you're going to find this gemmule formation inside the spongiocele. And these cells, which will form as conditions in the water become unfavorable, like if food were to become scarce, and this allows the sponge to survive until more favorable conditions return. It's almost like a seed pod, but they're not plants, so they don't actually have seeds. Hopefully you get the connection there. And another way that they asexually reproduce is through regeneration. When a part of them is broken off, they can regrow that lost part. And sometimes the part that broke off can grow into a whole new sponge, and then that loops us right back to budding. That's where part of it breaks off and grows into a new sponge. Just what you wanted to talk about with your zoology teacher, sponge sex. So some sponges are capable of having separate sexes. They have male organs that release sperm into the water, and then they have female organs that release eggs into the water. And then in the water, when the sperm and the egg meet, you have fertilization. The fertilized egg is a ciliated swimming larva. So it has cilia on the outside of it that allows it to move, and then it will settle down and grow into a whole new sponge. Finally, there is some hermaphrodism, which is where some sponges will have both male and female parts in the same body, so they can release both sperm and egg. With all the diversity of sponges, there are 3 different body types that sponges tend to have. The first kind we're going to talk about is ascon. This is the most simple body form, but it also happens to be the least common. So we don't see this very often. And you can see a diagram of it here in this bottom right-hand corner. This is where the water will come in through the in-current pore. It then enters the spongiocele that's lined, the inside of it's lined with collar cells to capture the food, and then the water leaves through one osculum. From an evolutionary standpoint, that ascon is the most simple and therefore probably the earliest body form that existed. Well, with time and evolution, we have the cycon body form. And this is where the sides of it have folds, and these folds serve the purpose of increasing the surface area, because We call these folds radial canals. And see how all the radial canals are lined with these black lines? Those black lines are meant to represent the collar cells. So we can increase the number of collar cells when we increase the surface area. And by increasing that surface area, that allows for more water to be filtered and therefore more food to be consumed. But we still maintain that flow into the incurrent pore. You get out of the radial canal into the into the spongiceal, and then out through the osculum. And with the cycon body type, there's still just one osculum. So the most evolved body type is the leucon. The leucon body type you'll find with most of the complex sponges, as well as in all the largest sponges that exist. This is where there are multiple oscula, and you'll see that in a second. So water can flow through any of these incurrent pores, any of these holes. Anywhere you see black, there are collar cells to grab any of the food. So you've got these chambers that are just lined with collar cells, increasing the number of collar cells, therefore increasing the amount of food that can be extracted from the water. The water often gets trapped in these chambers, and that gives the collar cells more time to get all the food out of the water, and then the water keeps flowing to one of the many oscula that exist for that particular sponge. Lastly, we need to discuss why sponges are important to mankind. Well, first of all, they're the basis of all animals. They're the most primitive, and from them, everything else has evolved. And so we're going to see certain characteristics of sponges that we'll see in other more developed animals later. But sponges provide homes for other sea creatures, and that's going to be important to the survival of the entire sea ecosystem, the entire ocean ecosystem. We used to use bath sponges. We used to use sponges from the ocean to clean our dishes and to clean ourselves in the bathtub. The reality is, when you go to the grocery store or any store and you buy a sponge, it's now not a living animal. At no point was it a living animal. They're made of synthetic fibers. But the sponges we buy are modeled after these sea sponges. However, you can buy some of these sea sponges in some stores, and people like to use them for painting purposes because it gives the texture to the plant, whatever it is that you're painting. We do conduct research on sponges, so that's important for scientists to continue collecting information about evolution and about sponges in general and that whole ecosystem that exists in the ocean. And finally, some people like to collect them for decoration. Not my style, but you know, different strokes for different folks.